Another biological reaction which I have done by the nucleic acids is protein synthesis. In this protein synthesis, two steps are involved. One is transcription. This transcription is the formation of RNA from the DNA. The transcription is DNA to RNA. And another one is translation. In this case, what is the RNA are formed in the transcription. From this information, the corresponding proteins are synthesized in the translation process. Now, coming to the mechanism of the transcription. In this process, DNA sequence into a complementary RNA molecule and DNA double helix partially uncoils. As shown in the picture, this DNA partially uncoils and one of the two strands act as a template for the synthesis of RNA molecule. Here, either of the strands act as a templates. Here, from the genetic information of the any one of the strand, here we take these are the base pairs in the strand of the DNA and the complementary bases are added to the corresponding chain and it forms the RNA which has a information about the DNA. That means each of the bases is added with the complementary bases, then the corresponding RNA is formed which has a similar sequence of the mother DNA. And after formation of RNA, this DNA again uncoils and remains as it is as the previous case. Now this migrated or synthesized RNA is go to cytoplasm. And this RNA is useful in the synthesis of the proteins in the translation process. Now, the RNA which is formed in the transcription process is called messenger RNA. Because this messenger RNA, the RNA which has a information about the mother DNA and it transfers this message to ribosomal RNA through the transfer RNA and the protein synthesis is happened at the ribosomes. And one of the strand, this means when this transcription process is going on, the RNA which is formed in this case has a information about the base pairs in the one of the strand of the DNA, that is information about the sequence of the bases. That's why it is called as messenger RNA. And this messenger RNA transfers the genetic information to the ribosomes through the transfer RNA. And at the ribosomes in cytoplasm, the protein synthesis is occurred. That is happened in the translation process. Translation. In this process, we have already discussed about the transcription. In the transcription, mRNA is formed from the DNA. But in the translation, protein is synthesized by using the mRNA sequence. Because from the mRNA, we can get the information about the order of the nucleotides in the corresponding DNA. Now, this mRNA is used in the protein synthesis. In generally, after transcription process, this mRNA moves to nucleus of the cell to a ribosome, which is present in the cytoplasm. Generally, nucleus has a cytoplasm, so the ribosome is present in the cytoplasm. So, this ribosome serves as a template for the synthesis of protein. This mRNA determines the order in which the different amino acid residues has to be joined. Because this rRNA has been synthesized from the DNA which has a genetic information. This genetic information gives the different amino acids in which order they have to be joined. The sequence of bases in mRNA are read in the serial order, in the group of 3 at a time. That means the sequence which is present in the mRNA is studied 3 nitrogenous bases at a time. The group of 3 templates or nucleotides are called codon. So total 20 amino acids present in our body. From each of these 3 triplets we can overall find that 20p3, that is approximately 64 types of codons in a different manner. Now here total 20 amino acids are present. So each triplet of codons represents the one amino acid. From these 20 bases we can represent the 64 amino acids. These 20 amino acids by group of 3 or triplet you can differentiate the 64 types of codons. It's like a 20p3, it gives the 64 types of codons. This code expressed in mRNA is read by tRNA and it is translated into the amino acid C. The function of the tRNA, it is express the code of mRNA by reading and it is translated into amino sequence. The function of here tRNA is read the sequence which is present in mRNA and the sequence is translated into amino acid. So that means this each codon, 64 types of codons, in each codon determines the one type amino acid. So when this tRNA determines the desired amino acid and transfer this information to the ribosome, and this process is repeated again and again, thus proteins are synthesized. When the synthesis of specific protein is completed and it is released from ribosome. This type of process is repeated again and again and it gives the total sequence of the amino acid, synthesize the corresponding protein. Now you can see this in the given picture. Here this is the mRNA which is produced in the transcription process. And this mRNA having a sequence of nitrogenous bases to the mother DNA. That means the genetic information which is having in the DNA is transferred to the mRNA in the transcription process. And these are the sequence of all nucleotides. And each 3 nucleotides here also called as codons. Now each codon represents the one amino acid. Here this is the ribosomes and here the protein synthesis is occurred in the ribosomes which is present in the cytoplasm, that is present in cell. And now this is the tRNA. This is tRNA and these two are ribosomes. These ribosomes are useful to protein synthesis and the information about the sequence of codons is transferred from the tRNA to ribosomes and the corresponding anticodons or corresponding amino acid sequence are read by the tRNA. And this information passes through the ribosomes. You can see this picture, it gives the clear information about this. One by one this sequence is read by tRNA and it gives the anticodons are formed in the surface of the ribosomes and the corresponding sequence of proteins is formed. This is the corresponding sequence of the amino acid is given by in this process and corresponding polypeptide chain is formed which is nothing but protein. And here after completion of the protein chain the ribosomes are released.